My name is Guilherme. I am a PhD candidate in genetics and plant breeding, supervised by Professor Labate. Now, I'd like to invite you to our last roundtable and the 9 MPB, entitled Integration of Transcriptomics, Metabolomics, and Ionomics, New Perspectives for Plant Breeding. This roundtable will feature 3 esteemed speakers, Dr. James, Dr. Felipe, and Dr. Douglas. After the presentations, we'll have a KI session to discuss these exciting omics themes. Talk 1, from breeders' ears to silicon ears, new tools for old challenges. Dr. James Snabel, University of Nebraska. I would like to introduce our esteemed speaker, Dr. James. Please, everyone, help me. Okay. Uh, Jamie is the Nebraska Corn of Presidential Chair and Professor in Department of Agronomy and Horticulture at the University Nebraska. He received his bachelor's degree in biology from Cornell University and his PhD in plant biology from the University of California. Has authored more than 130 scientific papers and leads measure-friendly funded projects totaling over $59 million. He also an entrepreneur, co-founder of startup Sork as Engineer's Egg and Diamond Genetics, and served as scientific advisor to companies Syngenta and Indoor Agriculture. James, audience, usual.
All right. Good afternoon, folks. Thank you for sticking around, and thank you. I don't actually see anyone sleeping yet, even though it's the afternoon. It's a warm day. We all had a very nice lunch. So I'm really happy to be here. Thank you to the organizers. I think Scott said it much better than I can, but this really is a remarkably well-organized conference, really enthusiastic students, great speakers, myself excluded. And most of the work I'm going to be talking to you about today is not my own. It's the work of the folks up in this photo, the members of my lab and my collaborators, and I'll try to call out who did what as I go through. If I forget, there'll often be pictures showing you whose project I'm talking about. Before I get started, the university, as you heard in my introduction, I've been involved in starting a number of companies. The university feels very strongly you should understand all my conflicts of interest. So, of these, probably the only one that you really need to know about is Dryland Genetics, which is a breeding company. I'll mention a little bit of work that the company has done later, but I've been involved in starting some companies, advising other companies. About 3 years ago, I spent 4 months as a paid employee of Google. With that, my background is I'm a geneticist. I want to know which genes do what. And there are a lot of genes in plants. In corn, there are 30,000 plus or minus 10,000, depending on who you ask. And the number of those genes where we really can point to, okay, we know what this plant is doing, or what this gene is doing in this plant. We know what the phenotype is of disrupting the function of that gene. It's very small. In maize, it's likely on the order of 500. Even in Arabidopsis, where researchers around the globe have spent decades studying it, it's a few thousand out of close to 30,000 genes. There are a lot of different tools for trying to figure out which genes do what. Classical genetics, reverse genetics. One of the most widely used are tools that use natural genetic variation, association studies, QTL studies. And my guess is a lot of you have either run GWAS or you've seen a paper that uses GWAS, but this is the very, very simple version. So genome-wide association study, we need a large, diverse population of some crop. Usually that's easy, depending on the crop. It may be hard just to get enough accessions. We need genetic information on every individual in the population. We need measurement of some phenotype of every individual in the population. And then we just do a simple association test. And you can think of this as something as simple as a t-test. We just add in a lot of control variables. We scale it up a lot. And we look for markers where there's a big difference in phenotype between individuals carrying different alleles of the marker. Sounds simple enough. And getting genetic information has gotten a lot easier in the 15 years or so that I have been doing plant science. The phenotype data remains quite hard. So we have conventional phenotyping, which is people walking through a field with rulers and notebooks, or maybe driving with a combine to measure yield. This has, if anything, gotten more expensive over time, because most of the cost of that is human labor, and human labor is getting more expensive much more rapidly than most things in our world. And more critically, the supply of graduate students who are willing to walk into a Nebraska cornfield where it might be 37, 38 degrees C, high humidity, blazing sun, and spend their summers working there, that's a fixed pool of people. We can't really get more of them. We want to use their time as efficiently as possible. So if we want more measurements of more traits and more locations, we're not going to get it from having more people walk through the field. And so then when I started as an assistant professor 10 years ago, people had identified this bottleneck and said, aha, we have a solution. We're going to invest in technology, robots, imaging, computers, AI. It's going to solve the whole problem. This is a great collaboration with one of my engineering collaborators, a guy named Li Tong, and that's one of my former techs, Lena Lopez. People keep getting hired away. It's a challenge. You get good people, and they rapidly go on and get great new jobs. Anyway, in 10 years of working with high-throughput phenotyping, the thing I'm going to tell you is it's not going to save you time. It's not going to save you money. What it is going to do is allow you to measure things that you otherwise would not be able to measure and understand things about plants that you wouldn't be able to understand before. Here's an example where we spent a lot of money and we didn't actually make very much progress. So one of the really challenging things about studying, say, genotype by environment interactions is we need data from lots and lots of different environments. As part of a large collaborative project, we were growing a panel of maize lines in 6 sites across 2 states. It takes probably 12 hours to drive from the easternmost to the westernmost of these sites, so you're not going to get to the sites very often. Even flying UAVs is a challenge. In the US, you have to have a drone operator who can see the drone, so they're not going to want to drive out to those sites. We wanted to test, could we use satellite data to measure properties of the plots? Now, we talked a little bit about that yesterday. It's possible. But we were using a satellite that provides us with 30 centimeter pixel resolution, so we actually have multiple pixels per plot. That is not a free satellite, it's a relatively expensive satellite. And we could train models to predict yield, predict flowering time from these satellite images collected during the growing season. The problem was that we can train the models within environments. They don't translate very well across environments. So this was an example of us trying to replace something that is slow and expensive and labor intensive, which is walking through the field to score flowering time, with an automated system. It didn't work so well. But I'm not going to just tell you stories about things that didn't work. Let's go to some examples where the high-tech methods actually help us. Now, this started out as another story where we were kind of discouraged. So this is work from Jensina Davis, a current PhD student in my lab, working with some collaborators at Purdue who work on 3D reconstruction. So we can take plants, put them through an imaging chamber, get back a 3D reconstruction of the plant. One of the things that allows us to do is actually measure the phyllotaxy of individual plants. If you looked at a corn or a sorghum plant or a wheat plant, I mean, you have this sense. I mean, the leaves come out one side to the other, and it should be very simple. If you look at actual varieties of the crop in the field or the greenhouse, there's a lot of variation. Some are really good at having perfect 180-degree angles back and forth. Some show a lot of variation. We'd like to understand that because that affects the dynamics of the canopy and light interception, lots of interesting properties. Nobody had really studied it before because it is a huge pain to measure. Think about being in the field, you have this plant, you're trying to measure the angle of one leaf relative to the next. So the best method we finally found to measure this by hand was to get the compass app on your phone, line that up with one midrib, then line it up with the other midrib and subtract the degrees. And so we did that. We made the measurements from the plants. We compared it to the computational measurements. That's what you see up at the top there. It was terrible. Correlation, R-squared is like 0.48. Less than half of the variation is being explained from the human is being explained by the automated system. So we could have again said, all right, that's it, we're discouraged, we give up, we're moving on. But we didn't, and I credit Jensina a lot for this. Jensina actually made those top measurements, and she said, well, how do we know it is the computer that's wrong? So she recruited another graduate student in my lab. They went back to the greenhouse. They measured the exact same plants, following the same protocol, one after the other. That bottom graph, that's the correlation between two humans measuring the exact same plants the exact same way. So almost all of what looks like error in the computational method, that's that humans are really bad at measuring phyllotaxy. So we're getting close to human level of performance, even with that quite low R squared. But that's a really complicated trait, right? I mean, it's something people haven't studied before. What about something simple? What about seed color? I work on sorghum too. I really like sorghum. You go to the field, you can see the diversity of sorghum right away in a way you can't with corn. So we'd like to study the genes that control the color of sorghum seeds. Sounds pretty easy, right? We had a population close to 1,000 varieties of sorghum grown in replication, harvested the seed, had undergraduates go through, score the seed. They had references of here are what the colors look like, and we had some seeds for them to look at, and they just had to score the seeds as one of 8 categories. And what you see here is the agreement between 2 replicates of undergraduates looking at sorghum seeds and scoring them as one category or another. You can see it doesn't work so well. Now, there are a couple of cases where I really don't know what's going on. It's either a data entry error or a planting error because we've got a black seed and then a white seed. But in the vast majority of cases, all we could get from the undergraduates scoring the seeds was, well, some seeds are light, some seeds are dark. That's about all we can consistently score from looking at a seed. It doesn't seem like it. I see color. A lot of us see some color, but we're bad at it. And if you take that light-dark scoring, you can do a genome-wide association study, as you see on the top, and you'll see, a couple of associations that line up with genes we know affect the color of sorghum. For example, the tan genes that are involved in tannin synthesis. But we can put those same seeds on a flatbed scanner. We can extract quantitative color measurements of the R, G, and B channels and the principal components showing variation in sort of light-dark. We can do a similar analysis, and we can find the same genes we found before with more statistical support. We can find additional signals. So even for things that seem really easy, that a human should be able to do, often the computer can give us a much more consistent, much more genetically controlled phenotype. And that's exciting to me. But the thing I want you to take away from this first little segment of my talk is that good phenotype data sets are going to remain rare, they're going to remain valuable, they're going to remain expensive, and part of our job as scientists, lots of other data is getting cheap, lots of other data is getting big, is to figure out how to use other types of data, other analyses, to make the most use possible of these phenotype data sets. This is an example of being able to make a new discovery without ever going to the field. What you see here are 2 genome-wide association studies conducted with the exact same marker data, with the exact same phenotype data for the exact same individuals, a large maze panel we grew in the field. The top panel uses the published set of markers from RNA-seq, about 1 million. The bottom panel uses whole genome resequencing, 40 million markers. And sequencing, I'd much rather spend money on sequencing than walk through a cornfield all summer. And we found a whole new gene. And in this case, again, it's a gene that we already knew about, ZCN8. It's one of the FT genes. But it popped out of the existing analysis just because before we didn't have a high enough marker density, we didn't have something in LD. But in these cases, I've been talking about genes where we already know what the true answer is. And genome-wide associations, even in species like maize that have very rapid LD decay, often will not give you that one candidate gene. Here's a zoom in on those 2 peaks I was showing you from the previous slide, and you can see that there are actually relatively wide regions of the genome that are being flagged as associated with our trait of interest. And the gene of interest is often not in the middle of that window. This is another example. In this case, we're looking at a range of photosynthetic traits measured in the field, so we don't know what the causal genes are. And again, this is work from Wakar, a current PhD student. Every one of those black triangles is an annotated gene, and there are a lot of genes in those mapping intervals. So many times, if you don't already know what the right answer is, GWAS isn't going to get you to the point of, aha, this gene does this thing. I wish it did. It would be so much more satisfying. And we can actually look at this more systematically. This is work from a former postdoc, Ravi Moral, where when we do know what the causal gene is, how likely is it that it's the closest gene to what the peak of our GWAS hit, the second closest, how far is it in space? And yeah, it's not perfect. So we need something we can do a little bit better than GWAS if I want to understand which genes do what. And that's what I want to do. I'm not a plant breeder. I'm a plant geneticist. I want to know what the genes do. And one of the, oh wow, the color really is a little weird. Anyway, one of the approaches we've had a lot of success with over the last 5 years is looking at transcript data rather than genetic marker data. Now, this does not intuitively seem like it would work, and it's really hard. So we're using the gene expression level rather than the genetic marker as our explanatory variable, but otherwise we're doing the same analysis we would do in a genome-wide association. This can identify important genes that were missed in GWAS, so whole new signals where you see no association from the genetic marker data. It is much less sensitive to linkage disequilibrium, as I'll show you in a couple of slides. But if I want to genotype a corn plant, an inbred line, I just need any piece of tissue from any plant of that genotype, and I'm good. And if I start with 500 lines and I'm not happy and I need another 400, I can just go and grow another 400. I can get the DNA. I can combine everything together. I can get a wonderful data set. With expression data, it's not so easy. I need the same tissue, but I need the same tissue in the same environment, and you can never really get the same environment back, so it needs to be the same tissue on the same day in the same place. And of course, the vast majority of genes in a corn plant or a sorghum plant or any plant exhibit diurnal cycling. Some of this is circadian. It's actually controlled by the circadian clock. But even if you're not controlled by the circadian clock, you're controlled by the fact that the lights turn on and photosynthesis starts and all sorts of things are fluxing through biochemical pathways and then they stop. So I need tissue from all the same plants on the same day in the same place, collected in really only an hour or two. And that's a really hard thing, and I think that's part of why TWAS, we haven't seen a lot of successes, is that's a hard bar to hit. Fortunately, I hired this guy, John Turkis. He's a really outstanding technician, came from a sort of construction background, and during the pandemic I said, look, John, we've got to do this thing. And he went away for a month, and he came back, and he built this whole system. There are some videos here, which I think I'm not going to be able to play from while standing here, but he's got these spill-proof trays, so you can flip over and your tubes don't go flying everywhere. He's got triple punches, so we're getting 3 sets of leaf tissue all at once, so when the first RNA extraction fails, we don't have to give up. We've got a backup, and when that one fails, actually, usually the second one doesn't fail. Then when somebody comes and says, oh, I want to measure ionomics, I want to measure metabolites, I say, hey, I've already got the whole panel sitting in my -80. So with the system John designed and a really motivated team of students, we were able to collect 2,500 samples in a period of only an hour and a half. That's 1 tube every 2 seconds. All right, so thank you. Isn't that nice? Have you collected things in the field? I've had tubes go spilling so many times. I mean, and this seems really simple, but it has, probably our data sets are 20% bigger because we have that chance to go back and try the sample again. With this expression data, we can do a couple of different things. We can try to connect, first we're going to talk about taking the gene expression as the explanatory variable, connect it to phenotype. So that's transcriptome-wide association. So here's that same phenotype data set I showed you before, where we went from million markers to 40 million. We went from 1 strong association to 2. This is Vlad's paper that Scott highlighted so kindly. I mean, I was really excited about this paper, and I did very little. All I did was convince people, okay, let's go to the field, let's get all this stuff, and then I recruited the right guy to analyze the data. So exact same phenotype data, genetic markers on the bottom right, and transcriptome-wide association is the big one. And you can see we get a lot more signals. Each signal is only a single gene. It's not a window, because we're not looking at the linkage disequilibrium isn't necessarily affecting the expression. A lot of the expression is reflecting regulation in other parts of the genome, transregulators. And these genes make sense. These are genes that are linked to flowering time. The vast majority of them. There are some that aren't, and we've got edits in the pipeline, and hopefully we'll find out if those are right or if those are false positives. But again, I want to know about new genes, and this is showing the method works, but we're finding things that we know about. So let me tell you another story using the same methodology in sorghum, my other crop, my other favorite crop. So the sorghum we grow in the U.S. is photoperiod insensitive. The sorghum that's grown in the tropics is photoperiod sensitive, so it'll flower in short days. Normal tropical sorghum in the US will flower in the winter and it will be dead long before that, so there's a mutation to remove the photoperiod sensitivity. But even after that, you are left with a lot of genetic variation in flowering time, and we know very little about the genes that control flowering time independent from the photoperiod sensing pathway. So we used the same approach, sampled 900 plots, 2 hours, RNA-seq on everything. And the nice thing about the RNA-seq is we also get the genetic markers for free. So we've got all the RNA, we can align them to the reference genome, and this population hadn't been genotyped previously, so we get some markers. Not 40 million, but a couple 100,000. So the first thing we can do is just run a genome-wide association study. This is using a method called farm-CPU, which is why you see single points instead of whole towers. And we get 3 signals. 2 of them are things that make a lot of sense. One's actually a photoperiod gene, Y1. The other is an FT-like gene, so like CCN8. One made no sense at all. It was just in the middle of an area, no annotated genes that have anything to do with flowering time. We'll come back to that one. Then we can do the transcriptome-wide association study. Again, we find lots of things, many of them that make sense. One of the things that we weren't expecting is we found multiple components of the autonomous flowering time pathway. Wait, not autonomous. Yeah, autonomous. Aging pathway. Sorry, there are too many flowering time pathways. Really, plants should be a lot simpler. But a microRNA that controls the transition from juvenile to adult leaves, and then that controls some transcription factors called either SPBs or SPLs, depending on what species you're working in. And then those activate another microRNA called miR-172. So we've got our miR-156 is showing up, our SPLs are showing up, miR-172 not showing up. The microRNAs 156 we detected, it was really nice because we realized that microRNAs aren't annotated in the sorghum genome. So this was showing up as some weird protein coding gene with no homology to anything. And once we realized that, we went back and said, okay, well, we're finding most of this pathway, but not the whole pathway. I wonder if there's anything else going on here. And so we went back to that, mystery hit on chromosome 9. And if you dig in, that is right on top of an unannotated microRNA encoding miR-172. So we are identifying candidate genes, even in cases where the gene isn't annotated, and we're pulling out this pathway that appears to play a large role in flowering time in sorghum once you remove the ability to sense photoperiod. So why does this work so well? It's a little strange. We have, thank you, 2 potential models. One is that, as I mentioned a little bit earlier, the expression of a gene doesn't just reflect the haplotype of the gene. It reflects the integration of many small effect variants upstream of that gene in the pathway. And so any one of those variants might have too small an effect on the phenotype to be identified in a genome-wide association study. But if they all feed forward into one integrating signal, we may pick up the signal of that gene through a transcriptome-wide association study, even if we're missing the individual transregulators in a genome-wide association study. The other thing is that transcript data is continuous. Genetic marker data is not. In highly polyploid crops, it starts to get pretty close to continuous, but in diploid crops, any one marker is only going to tag 2 functionally distinct haplotypes. So by having expression data, if there are 3 or more functionally distinct haplotypes segregating in our population, we're able to recover much stronger, better signal than a marker that's only going to ever distinguish 2 of those 3 haplotypes from each other. But, okay, I probably should have presented those in the opposite order. We are really interested, though, in this idea that what we're finding is the integration of multiple upstream regulators that all feed forward into the expression of one gene, and that's why that gene is coming up in the TWAS, even though there's no genetic association from the location of that gene. And the really nice thing with having this expression data across large populations is we can take the expression data, instead of using it as our genotype, we can use it as our phenotype. And now we're using genetic variants to explain our phenotype. We're using gene expression as a phenotype. We call this EQTL instead of TWAS. I don't know why that one's a QTL and the other ones are association studies. The literature doesn't always make sense. But we can do an analysis like this. This is a particular gene of interest. You can see that the genetic variants close to that gene explain a lot of variation in expression. So that's a cis-EQTL. It's likely a variation in the promoter or the regional context. We also see a very strong signal from somewhere else in the genome. So this is a transacting QTL. It could be a transcription factor. It could be a microRNA. It could be a duplicate copy of the gene that's present in some individuals but not others, but it's not genetically linked to the gene itself. And because we have the expression data from both maize and sorghum, we can run this on all 20,000 expressed genes in the sorghum plant and the maize plant. And we can line up where are these transregulators across the genome. When we do that, the top panel here is maize, middle panel is sorghum. In maize, there are transregulators strewn all across the genome, sort of at random. In our sorghum population, there are just 3 loci that each individually control the expression of more than 300 genes in the leaves of sorghum plants in Nebraska in the summer of 2021. I don't know if that will translate or not, but at least in that environment, these 3 guys are making a lot of the calls. And we can, what you see on the bottom is just where the specific GWAS hits are lining up in the individual genes, and they're sitting right in the middle of 3 genes. And I think I'm running out of time, so we're going to tell this story pretty quickly. In at least one case, it turned out this was sitting in the middle of the sorghum ortholog of a Arabidopsis transcription factor called Hi5. And we were able to demonstrate the genes that were being regulated by this were disproportionately likely to be the orthologs of genes that are regulated by the Arabidopsis Hi5 transcription factor. So that gives us confidence this is working. Also, once we know this, we can use a phenome-wide association study, apologies for all the association studies, where we just take that one genetic marker and we look at phenotypic variation, and we can start to map out how that marker, identified purely from molecular phenotypes, is affecting the overall state of the sorghum plant as an organism. I was too ambitious. 5 minutes? 3 minutes? 5 minutes. Okay. Yeah, I think we'll end with this story. I had more stuff. One of the other things with having a data set like this is once you have it, people come up with really weird ideas for how to use it. So I had a undergraduate, Charlie, joined my lab over the summer. We were talking about this data set, and he said, well, could we measure the bacterial content from the RNA-seq? And I said, no. We poly-A select, so we can't, the bacteria RNA will not be in the sample. He thought about that for a little while and came back. What about the fungal DNA? So I said, I don't think that's going to work, but you're here for the summer. He developed a pipeline. He went in, he pulled out just the fungal reads from every one of these samples. At this point, we had sorghum data, we had maize data from 2 environments, and in every case, he got thousands of reads. We started out with 10 million, so thousands is very little. Thousands of reads that map back to fungal genomes, and he was able to quantify the abundance of individual fungal taxa in each of these samples. If you see that sort of brown splotch over there, those are the samples that show high abundance of Puccinia, which is the causal pathogen for southern corn rust and common rust. And in fact, we had an outbreak of southern rust in the field that year. So we could line that up a little bit. But we wanted to know, does this actually work? Does this actually tell us anything interesting? And this is one of the examples where we actually found something that translates into new environments. Hopefully, yes. All right, so first, what you see on the far side of the slide is looking in 2 different environments, Nebraska in 2020 and Michigan in 2021, doing genome-wide association studies for the abundance of different taxa. And what you see is in many cases, we identify the exact same position in the maize genome as associated with the abundance of the exact same fungal taxa. This is usually at a genus level, not at an individual species level. So that shows that these are at least consistent, but are they affecting anything we care about in the plant? What you see on my side of the screen, and this is work from a brand new PhD student, Carla, she started in August, is that if we take the loci that were associated with the abundance of a different pathogen, I'm not a plant pathologist. Anyway, the pathogen that causes anthracnose in sorghum, and we take that from 2021, and in 2025, this year, we score the severity of anthracnose in the same population in the field. The signals we get line up perfectly. So by measuring fungal abundance, something we never even planned to do in the field, we were able to identify loci that translated into disease severity or resistance in a new field. And that's pretty exciting. Now, I think I talked slower than I thought, so I'm going to skip over this whole story about other stuff and say thank you all very much for your attention. Thank you again to the organizers. Thank you to these great folks who actually did all the work I've been telling you about. And yeah.
Thank you so much for your lecture, Dr. James. Your talk was excellent. And now you may have your seat in the audience. So let's move on to the next speaker. To continue our roundtable discussion, I would like to introduce our estimated speaker, Dr. Douglas Silva Domingues. Douglas Silva Domingues is an assistant professor of molecular genetics at Exalc. He graduated in biology at Unesp Botucatu and earned a PhD in biotechnology from USP with a doctoral internship at CIRAD, as on sabbatical leave at the UC Davis, USA. He leads a research group in plant genomics and molecular biology focused on carbohydrate and terpene metabolism, the evolution of gene families, transposable elements, and no-code RNAs, and gene expression under stress using multi-omic approaches. Dr. Douglas, the audience is all yours.
First of all, thank you for the organizers for inviting me in the last moment. I am replacing here Dr. Danilo Dalloso, who is a great guy in metabolomics. I'm not giving to you like 10% of the talk that Danilo could give to you here, but I'll try to do my best over here. So I'm going to tell you a story about my my favorite system as the previous talk told to you. I'm also a plant geneticist. I like to know what genes are doing in plants, and for him, his favorite models were maize and sorghum, and my favorite model is coffee. So it's not, I do not need to spend a long time here, but I think that most of you like to drink coffee. Most of you to be awakened in this talk here need to drink some coffee too, but so Brazil is the most important producer, and these regions here in Brazil are the most important producers in terms of Arabica coffee. So one thing that's nice to be told is that Arabica coffee is a polyploid, it's a tetraploid, with two potential parental species, Coffea eugenioides and Coffea canephora. And just to make a long story short, what I'm going to talk here to you guys is basically about RNA-seq profiling, and couple a little bit of this with chromatography and mass spectrometry data. We had advanced quite well in transcriptome analysis, not that well in metabolite data. So I'm going to talk basically about these to you using Arabica plants. So for this, we are going to compare two Arabica varieties, and that's a little bit where my story links more well with breeding programs. So in the case of Arabica coffee, I'm not going to tell you about all the history of coffee breeding, but to make a long story short, we can roughly divide coffee Arabica varieties in pure and introgressed varieties. So the first varieties that were released by Brazilian programs in coffee breeding, they are known as pure varieties because they are basically crosses between historical genotypes that we are using in coffee plantations. They are still the most important ones in terms of area in Brazil. The good part that they are historical ones. The bad part is that they are prone to most diseases that are common in coffee, namely rust, brown eye spot, and other ones. As you can see here, in my case, I used Catuai Vermelho as a pure Arabica variety. It was released in the 70s. And as a counterpart, I am using Obatan. That is an example of an introgressed Arabica. So these varieties were released from the 90s, in the case of this one, the year 2000. And differently from the pure Arabica ones, they came from an introgression with materials that were mostly from Asia that carries alleles from other coffee species, and they are widely known by the coffee breeding community as introgressed Arabica varieties. The good part of these varieties is they are more resistant to the most common diseases in coffee. And you can see that besides the differences in resistances and susceptibility, they also have differences in fruit ripening time, as you can see in this comparison here between Catuai and Obatan. So here I'm going to talk with you about the effects of plant, a substance that can be used for a priming effect in plants. So just to put everybody on the same page, plant priming is basically a process that you can expose the plant for a mild stress, usually using a chemical compound to do this, because then the plant gets like a memory to when we have another stress, and it's easier to react against this stress after this priming, which diminishes the impact of the uses of other inputs in plantations. In my case here, I'm not going to challenge coffee with any pathogen, but I was more interested in understanding the effect per se of these priming substances. And the one that I used in this case was xanoic acid. And why did I choose this one? Basically because we had prior results in citrus and tomato as a good substance priming well-known pathways in tomato. So with some colleagues, we also tested it against citrus canker with good results. And we also had tested it with in vitro with some common coffee pathogens with good results too. That's a little bit why I chose this one to delve a little bit into in terms of transcriptome and metabolome. So the central question that I'm going to tell you the story over here is how does this priming agent, xanoic acid, could modulate the transcriptome and the metabolome of those two varieties, Catuai vermelho and Obatan. So this is the very common process of obtaining this data. So we did RNA-seq data with mature leaves and roots of these two genotypes that were growing nutrient solution, and then we added xanoic acid, and then after 48 hours we collected the samples for leaves and roots, and then we extracted RNA, we did all the RNA-seq analysis, and we also extracted metabolites from the same samples, and we did GCMS analysis of these same samples in order to compare the responses between those two cultivars. So in terms of transcriptome, so in leaves we have very few differentially expressed genes in response to this priming substances. Here are the most important ones. So we have just 8 genes in common between those two varieties that are differentially expressed. So most of the genes are specific to each cultivar. Then I give you some examples of genes that are commonly regulated in those two genotypes, because they are so few that I can put this in a table. But most of the genes that are regulated by the using of this primer are completely different between those two genotypes. So going further into the most important differentially expressed genes in those two genotypes, you can see that the most differentially expressed one is the same one for the two genotypes. However, if you go further in this list, then you go that you have completely different pathways that are regulated between those two genotypes. For instance, terpene pathway is upregulated in Catuai, but not in Obatan, for instance. So by the other side, caffeine synthase is upregulated in Obatan and not in Catuai, for instance. So giving a more broad analysis of this data, we can see that some processes are quite different in regulations when we compare those two genotypes. For instance, primary metabolic processes are a little bit more regulated by the application of these priming substances in Catuai when compared to Obatan. And also we have some differences in nitrogen compounds and organic substances. So We also have some cases of specifically genes that were regulated just in one of those two cultivars, like cellular responses to stimuli, responses to stress, just in the susceptible variety, which came to our attention that only the susceptible variety has activated those pathways after priming. So we also analyzed the roots, as I told you. So in the case of roots, since we are applying the primer directly more closer to roots, of course, we have more differentially expressed genes in this case. But again, we have a panel much bigger of cultivar-specific differentially regulated genes rather than common genes. And in this case, we can see that Obatan regulate much more genes than Catuai. And what came to our attention is that Obatan usually down-regulates genes when we apply the primer, and Catuai usually up-regulates genes in this case. So we also made the comparison of the process, and we also have specific processes in Catuai, and we can see that some processes are quite upper-regulated in Obatan when we compare it to Catuai. So we also made a table with the most important genes in this case, and then we have a completely different scenario when we analyze roots of Catuai and Obatan. So genes that are mostly differentially expressed between those two varieties are completely different when we apply the same primer. So in terms of transcriptomes, we have clearly distinct differentially expressed genes, and in the case of Obatan, we have more defense-related pathways that are upregulated. In the case of Catuai, we have most basic metabolic adjustments. So then we take a look at the metabolome. So we have much less metabolites when we compare it to transcripts, with it's kind of expected. But differently from what we saw in transcriptome, we have much more shared metabolites between those two varieties. Differently from what we see in terms of transcriptome. So we have a very few differential metabolites and a lot of different transcripts between those two varieties. So when we analyze, for instance, in leaves, we have cases that we have completely different profiles within those two varieties. In this case of the 2-oxogluconic acid, so we have down-regulation in Obatan, as well it's quite good with a quite good quantity within Catuai. So in terms of different specific metabolites for those genotypes, in the case of ethanoic acid, we do see a difference in upregulation of arginine and some compounds of the primary metabolism. So in the case of Obatan, we have also arginine, and we also have caffeoylquinic acid. That's a very well-known chlorogenic acid related to coffee quality. So when we see the overall metabolism in roots, we again have a more common path of common metabolites and quite a few specific metabolites, which are also different from transcriptome data that we had before. So in the case of differentially abundant metabolites, we again have a very few metabolites that are specific to each phase. And the metabolites that are differentially between Obatan and Catuai, they are different than I'm going to show you here. So the metabolites that we have more in abundance in Obatan, most of them are related to lipids. And in the case of Catuai, most of them are related to carbohydrates. So in the case of metabolome, you can see that Obatan has a more lipid-like response to the primer, ethanoic acid. And in the case of Catuai, we have more carbohydrates that are differentially regulated after the application of ethanoic acid. So we can see that with less data points, because we see much less differential abundance in metabolites, but we can see clearly distinct responses even with that in terms of lipids and carbohydrates. So giving an overall profile of, I showed you here, guys. So we see, of course, greater plasticity in roots, but this is a kind of expected since we apply the primer in roots. But we could see a difference between Catuai and Ubatã, where Catuai has a preference to accumulate carbohydrates in response to this primer, and Ubatã a more abundance of lipids. But we do see a systemic modulation of the plant metabolism much higher in transcriptome when compared to metabolome. So the metabolome, the transcriptomic part of this history is already published. Now we are working on the metabolome data in order to make more correlations between transcripts and metabolite data in order to understand more this huge difference between transcripts and metabolites. But we are able to see that we have a good story over here to be counted. So basically why this kind of analysis matter for breeders, as you can see, you have a completely different responses applying the same primer when the genetic background is distinct between coffee varieties. So it's important to apply multi-omics strategies in order to better depict best candidates as being told by the other speakers here. So with this, I'm finishing my talk here. So I'm going to show you an all-girls team that worked in this project. Ilara, Paula, Raissa, Natalia, and Susana. So I had an all-girls team working on that. All of this metabolome data was produced with the collaboration of Professor Carlos Labate. And of course, we need to say who are the guys who are paying all this stuff. That is CAFAPESP and CAPES. And with this, I would like to thank you.
Thank you very much, Professor Douglas, for your excellent speech. And now, to continue our roundtable discussion, I would like to introduce our next speaker, Dr. Felipe Hikashinevsky. Please, everyone, help me to say hi and welcome to Dr. Hikashinevsky. Dr. Hikashinevsky is an associate professor in the Department of Botany and at the Federal University of Rio Grande do Sul, where he also contributes to the graduate program in cell and molecular biology and in botany. He holds a bachelor degree in biological sciences, a master's and a PhD in cell and molecular biology from Federal University of Rio Grande do Sul, with doctoral training at Dartmouth College and research experience in leading laboratories such as Purdue University, Brookhaven National Laboratory, Stanford University, and University of Aberdeen. His research investigates how plants regulate their genome, focusing on nutrient transporters, ion homeostasis, and strategies to enhance nutritional quality and efficiency in crops like rice and Arabidopsis. Dr. Hikashinevsky combines functional genetics, bioinformatics, and synchrotome-based approaches on his studies. He has been recognized with several honors, including support from the Serrapilheira Institute, election as affiliate member of the Brazilian Academy of Science, and Fulbright Fellowship in 2022. Dr. Hikashinevsky, the audience is all yours.
Yeah, thank you. Thanks for the very kind introduction, Raissa, and I want to say exactly the same thing that everyone said in this meeting. This is by far the most well-organized meeting I've ever been to. You were all excellent in doing that. We feel very welcome, and it's really good to be here, so thank you very much for that. You have a lot to teach to us, a little bit older, that also organize meetings. I'll be back to get some tips from you, so thank you very much. So, as Raissa mentioned, I'm Felipe. I'm in the southmost state of Brazil, in Porto Alegre. If you ever have the opportunity to come by, please say hi and go visit my lab. And I'm going to be talking about the ionome, which is my passion. That was mentioned a couple times here, so I'm very happy to see it. But I always like to, let me see if this is working. Oh, I think it's not. So I think you have to put in the adapter. So if you may do so. Yep. I don't think it's going to fit, so you have to put in the, if you want some help, let me know. I'm the last one, so I'm sorry for that. So, nope. Yeah, this is where. Excuse me, just a minute. Sorry for that. So, okay, so I think maybe I'm too far. Oh, there it goes. Okay, so I always like to define the ionome. I think you guys probably are very interested in omics, so you probably already can guess what that is. But I always like to define as the inorganic elemental composition of any organism that you want to study. And of course, you can always think of the ionome of parts of that organism. So for example, we can work with the ionome of the seed, the roots, the leaves, or even single tissues, cells, or organelles. So we're really looking into the detail of the distribution of different ions within a single cell of a plant, for example. So why I think this is important and not only another fancy name for us, instead of doing elemental profiling, we're calling it ionome. We think this is a very important concept because we're very used to, and we achieved a lot by doing experiments when we actually vary one element in our experiment. Let's say we are growing sorghum plants, and then we have low phosphorus, high phosphorus, and we measure phosphorus, and it correlates with what we did with the plants. But we sort of don't look at the other stuff because I'm changing phosphorus, so why do I care about nitrogen or iron or zinc? But we know that actually those change when we change phosphorus. And the reasons why that happens is not because the plant is a passive thing that just accepts that because they have some connections in the soil, but actually the plant regulates that balance using molecular mechanisms that we don't know of. So that's what we want to do. We want to understand that. So that's why we try to see and measure as many elements in any sample we can get our hands on. So the reason why we're very interested, I'm going to show you one quick example here in Arabidopsis. I think I'm too far. Okay, that works. So this is a quick example in Arabidopsis. Arabidopsis is a model for us. So let's say we're doing an experiment where we put Arabidopsis plants in low iron and also in control conditions of iron. And I ask one of the students to measure iron in the leaves. Probably you're going to get, after 2 weeks, samples, and you expect the samples from this plant to actually be low in iron. But when this experiment is done, this is what you get. So this is like 10 times less iron than this control plant, and you can see that the concentrations of iron are just the same. So iron is not changing when you put them in low iron, but it is expressing a high affinity transporter for iron and also a reductase in the roots. So the plant is responding to iron deficiency, and the response is being efficient to maintain the levels of iron. So the student was not wrong. We actually are not seeing any change. But since we measure as many elements as possible, you can see here that other elements are actually changing in iron-deficient plants. So you can see that manganese, cobalt, zinc, and cadmium all go up, and molybdenum goes down. So we know the reasons for that. We're not going to get into that detail, but these elements are telling a better story about an iron-deficient plant than iron itself. So we have to measure as many elements because the ionome indeed is integrated, and it's important to study that way. So why we're interested in the ionome in the first place? First and foremost, we want to do biofortification. You probably heard about golden rice, which is for vitamin A, for beta-carotene. We want to do something similar for iron and for zinc, which are our favorite elements. And also we want to make food both more safe and also more efficient in acquiring nutrients from the soil. So we have to regulate properly the ionome. So I'm going to try to tell 2 stories from my lab, and the first one is involving Arabidopsis thaliana. I know Arabidopsis is a model also for breeders, but it's not the most interesting things to you breeders, but I'm not a breeder myself, so I'm going to try to show you that this is interesting for some reason. So what we're trying to understand here is not only the ionome of Arabidopsis. We want to know which genes are controlling this ionome, but not in any Arabidopsis accession. We're using natural variation of Arabidopsis. It's not a plant that we eat, so it grows in the wild. We can go there, collect samples, and see how that is being regulated in different accessions. So this is what we're using. We're using ecotypes or accessions of Arabidopsis that are spread around the Arabidopsis diversity where it grows naturally. So I think this is the first time I actually don't have to, I feel like I don't have to explain what the magic lines are. So I'm not going to embarrass myself in front of all the breeders and try to explain as a biologist what that means. You know better than me. If you want to talk about that and teach me about it, I'll be very happy to learn from you. But these are this is the magic population for Arabidopsis. It used 19 founder accessions. They mixed up all the genomes of those founders. They're very widespread in the diversity of Arabidopsis. And after 4 generations of outcrossing, they got more than 1,000 lines, after selfing them for 6 generations. And more than half of those were fully genotyped using SNP chips. So we kind of know which part of each chromosome is being contributed from which of the parents. So we can do mapping with that. You guys know that better than me. So enough of my breeding. I don't want to embarrass myself. So what we did is get all of these lines and do ionomics profiling with them. We grew in the same environment, in the same greenhouse, everything very controlled, nothing in the field, very, very, very well controlled. So we want to know the genetic contribution to that. So we measure more than 20 elements. And of course, we can do analysis with that and see which regions of the genome are contributing to the variation that we find. Again, I didn't do that. This is a very good collaboration with my mentor for a very long time, David Salt, and also Paula Cover from University of Bath. We just recently published that paper. So we got a lot of interesting stuff, but they were all published by David itself. I mean, he had published that before, but okay, we want to see something new. You mentioned the same, right, James? So we want to see something new. And luckily, when I arrived in David's lab when I was a postdoc, actually we found one QTL for zinc, which is one of my 2 favorite elements. The other one is iron. So we found that very nice peak right there. And we can see that one of the parents is actually contributing strongly to a high zinc phenotype in the leaves. So now I'll stop with the breeding and now I'll go to the functional genetics, which is what I know how to do. So we got very excited about that because there's one gene that is doing something there to increase zinc concentration. We have 3 different experiments done in 3 different labs across the world by different people, and they always show sort of the same. When you have the parent KN0 and Columbia, which is the reference in Arabidopsis, we always have high zinc in KN0 compared to Columbia. The percentage changes, but it's always the same result. And interestingly enough, if you're thinking about biofortification, we're not going to eat Arabidopsis anyways, but it's interesting that whatever mechanism is working is actually getting more zinc into the seeds. So that's also interesting. So we want to run after that gene, and within that interval, that was a very good candidate. So we were doing exactly what you're showing, James. There's a lot of genes, but there's one good candidate, and that one is MTP3. So now I'm in familiar territory. This is a transporter, and we kind of know what it does. So it's a transporter that every time the roots get more zinc, there's excessive zinc in the roots, it comes up, and the transporter is in the vacuole of the roots. So it avoids too much zinc going into the xylem and therefore into the shoots. So that kind of makes sense. So maybe KN0 has high zinc because it has either a hypofunctional or a non-functional or a low expression allele of MTP3. Because if it's not working too much, you don't have as much zinc being locked, being detoxifying the roots, and then more is going into the shoots and into the seeds. So we got very excited about that and decided to go after. So the first thing that we do in functional genetics is try to complement our lines. So what you're seeing here is Columbia, which usually has the lower zinc concentration in leaves, KN0, which is the other one with high zinc, and these are lines that are actual KN0 plants, but we put back the genomic copy of MTP3 from Columbia. So we switch, so we got this copy, an extra copy of this gene into this plant, and now we got low zinc concentrations. So that shows that this gene is actually causing the phenotype that we've seen of high zinc concentration. So we're very happy about that. MTP3 is indeed causing the phenotype we found. We want to know exactly which mutation was doing that, so we searched for SNPs that were exclusive for KN0 compared to the other founders in the Magic Lines population. So we found one SNP. We got very lucky. Arabidopsis sometimes is very nice to us. So this is the one SNP that we found is in the coding region of that gene and actually causes a very nice change from an isoleucine to a lysine. That's a very strong change in the sequence of the transporter, and that is in the 6th transmembrane domain, which actually touches, which makes contact with zinc, so it's probably doing something. We don't know exactly what it is, but it kind of makes sense. Of course, we want to do functional thing, so we did that with yeast complementation. So I'm going to walk you through that kind of assay if you don't know it. What we're seeing here is a wild-type yeast in diluted series. So these yeast usually grow well in those plates. And here, the three other lines is a mutant of yeast. So these mutants cannot detoxify zinc into the vacuoles. So they're growing all fine here, and this one is the mutant, the pure mutant. There's nothing, we didn't insert anything in the mutant, so if we increase zinc, it's gonna die. But here, we inserted the Columbia copy of MTP3, which we think is fully functional, and the KN01, which we think has something wrong with it. So they're growing zero in regular zinc, so they're all fine. And I'm gonna call your attention to here when we have 1 millimole. This is a lot of zinc, but it's just to prove our point. So what you're seeing here is the wild type growing well, the mutant is not very happy, but when we put either MTP3 from Columbia or KN0, now they're fine. They can complement the phenotype of the yeast, which is showing that they can do the same function at detoxifying zinc into the vacuoles, right? But I'm gonna call your attention to now 10 times more zinc. This is a lot of zinc. But what happens when we do that is wild type is fine, the mutant is not happy, and the mutant complemented with the Columbia copy now is happy, but the KN01 is not. So this is telling us that probably that copy of the gene is coding for a protein that is hypofunctional. It is able to detoxify zinc, but only up to a point. If we increase zinc too much, it doesn't work as well. So that fits nice with our hypothesis. So it's probably what we call a weak allele, or you probably guys know more about how we name those things, but that's exactly what we're excited about. So this is our current model. If we think about zinc going radially from the rhizosphere into the xylem, going through all the layers that we have in the Arabidopsis root. In Colombia, usually zinc gets into the symplas. A little bit of zinc is being detoxified in the root vacuoles, and then we have some amount of zinc to be loaded into the xylem and then up into the shoots. That happens in Colombia, but when we have KN0, we have, again, zinc getting into the symplas, but very little zinc is actually getting inside the vacuole because the transporter is not as efficient. So more zinc is actually being loaded into the xylem, which increases the concentration that we see in the upper tissues, both shoots and also in the seeds. So that's the model that we have thus far. So this just came out. We just came out with that paper, so I'm very happy that it got accepted. If you're interested, you can take a look. It's out and it's publicly available. So now I'm going to switch gears and go to the second story. So now we're not doing breeding anymore. What we're going to do is straight candidate gene approach for biofortification in rice. So you guys know that rice is a very important crop. It feeds more than half of the world's population, but it's a very low source. It's a very good source of carbohydrates, but not a very good source of other nutrients, such as my two favorite ones, which are iron and zinc. But the problem is that iron and zinc, they're my favorite ones, but they're very important to everyone. So we know that a lot of people, they have problems with iron and zinc deficiency, and the problem with rice is that it doesn't have as much iron and zinc as we wanted, and we make it an even worse source of both of those nutrients, because we tend not to eat the brown rice. Usually guys take a look at the brown rice and go for the white rice, which is the polished version of brown rice. We get rid of the external layers, which are the ones that actually have some iron and zinc. So it's not good anyways, and we make it even worse by eating white rice. There are other reasons for that, but that's what we do. So we have very little in the tissue that we eat, which is the endosperm of the seed. So this is a problem because iron and zinc are very widespread deficiencies in the human population. We're in the estimates of billions of people suffering from that in the world. And even in Brazil, now we have some estimates that 1 in every 3 kids under the age of 5 in our country actually suffer from iron deficiency. We always thought that this was a thing from the other places, but even in Brazil, this is really bad. Even if the estimates are a little overestimated, this is really bad. So we need to solve that problem somehow. So what's our very simple goal? We want to increase iron in the endosperm, in the seeds, and particularly in the endosperm of rice plants. So this was done with golden rice. People increase the vitamin A concentration in the endosperm. We want to do the same with iron in the seeds. Of course, it's not going to look red like that, but I just wanted you to remember what we want to do. So we want to change the concentration of iron in the endosperm of rice. So the good thing is that we do have a family of genes that are good candidates for doing biofortification. So this was published a number of years ago, almost 20 years ago, by Mary-Lou Guérinot, one of the best mentors I've ever met in my life. This is a science paper, and they describe this family in Arabidopsis called the vacuolar iron transporter. So it's one copy gene in Arabidopsis, and what they did, they mutated that, they got a mutant for that gene, and what happens when you mutate that gene in Arabidopsis, the concentration of iron in the seeds does not change, it's exactly the same, but the distribution of iron within the seed is completely changed. So you can see here iron is always associated with the vasculature, both in the radical and in the two cotyledons of Arabidopsis. And when you mutate VIT1, this is the wild type, of course, when you mutate VIT1, you have everything kind of scrambled, and actually iron starts to be located where manganese is. So you can see here the localization of iron and manganese is the same here, but it's not here in the wild type. So we have this effect on distribution of iron. So everyone was very interested in thinking what would happen in a cereal that we eat. Maybe we have a change and we have more iron in the endosperm. So Jimmy Gong did that also more than 10 years ago, and they mutated the VIT copies in rice. But in rice, we have two copies, VIT1 and VIT2, and they only got, those were the tDNA years. We didn't have CRISPR-Cas9, so they got different mutants in different backgrounds, so they could not get a double mutant. And when you mutate those two genes, what happens is that luckily, not only the distribution of iron changes, I'm gonna show you in just a minute, but the concentration of iron increases in the seed and also within the endosperm. So what happens is that you have this change of distribution. Here's the wild type, VIT1 mutant, wild type, VIT2 mutant, but also the concentration in the whole seed and also in the endosperm is kind of increasing. And later, Jianfeng Ma published this one paper only doing CRISPR-Cas9 in VIT2. Actually, we got scooped because we were trying to do that and we didn't publish, so he got that before us. So what they've shown with this paper is that you can see here the iron localization in the wild type. You don't see much in the embryo, but when you mutate VIT2, you have much more iron in the embryo, and you have a change in the aileron layer. So this is the wild type. You have the external layer of the brown rice. They have some iron, and now you get less iron in the mutant in the aileron layer, but more in the endosperm. So that was very encouraging for us. But what we're asking is that since you have two genes and no one has ever generated a double mutant for those, what would happen if we do that? So we decided, at that time we had one undergrad student with no fellowship, and she said, yeah, I'll do it. So she got one vector, that was 2000 something, and we only could do one guide RNA. So we got one for both genes, and she got a good hit, so all the lines were double mutant for both genes. So I'm gonna show you just a minute. This is what we're asking, and this is failing, sorry. Oh, there you go. So this is Felipe Marasquin, my great collaborator. We share the lab. We like that we have the same name so people get us confused. And this is Bettina. She's an amazing student. As an undergrad, she generated those plants, and we got 5 lines that were mutated for both genes at the same time. So that was very lucky, but of course, the first question they would ask, like, do those plants have iron in their seeds? And luckily for us, they do. So these are 2 greenhouse experiments. We have 2 lines compared to the wild type and 4 lines compared to the wild type. You can always see that the iron concentration is higher in those seeds. Zinc is also increased, so we got lucky for both of them. We also have some changes in other elements, but they're not as consistent as iron and zinc, so it seems to be kind of specific. Molybdenum is the only one following them. We don't know exactly why, but that's what we see. So we were very happy to see that we actually got increased iron concentration, but we wanted to do more. So we, of course, we're doing functional genetics, but we also in the plant physiology lab, so we wanted to understand more where was that extra iron coming from. So we partnered with some collaborators in Italy, in Bolzan Free University, and we did one experiment with heavy isotopes of iron. So the way we do this experiment, I'm going to try to show in a simple way. So we can feed the flag leaf, which is where the photoassimilates come from to fuel the seeds when rice is increasing its grain filling. So we can feed the flag leaf with a heavy isotope. This is a low concentration, so we can make a cut and we put that leaf into the iron solution. And we can do that with a low concentration and also with a high concentration. And to compare, we did that with roots as well. So we feed them with a low concentration from the roots, and then we can measure that heavy isotope in the seed to see where the extra iron is coming from. And these are the results we got. So we can see that the low concentration in the flag leaf, we get very little of that in the seed. If we increase the concentration in the seed, everyone gets higher concentration, but we don't see a difference in them. But when we feed through the roots, now we can see that we have more of the heavy isotope there. So what's happening is that the extra iron is actually coming from underneath, from the roots. So of course, we wanted to know where exactly that iron is. We didn't show that for our double mutant. So we partnered with some collaborators here from Sena, from Piracicaba. So I have an amazing partnership and collaboration with Dr. Hudson and his team. They've been helping us a lot here. So they did that experiment right here. So what we did here is to section the seed in half, and then we can do a line scan using an X-ray fluorescence equipment. It's a benchtop one. So when you do a line scan, you can actually get signal from iron. So what was the expectation? We would see high iron in the corners, which is the aileron layer, which has high iron. And then when you go inside the seed, you see almost no iron. And our expectation, if the endosperm had increased iron, is that that gradient would be less steep. So it goes down not as fast as the wild type. Here, it seems that that's true, but of course, you're not going to just believe me because I'm showing a pretty picture. We have to quantify that. We did that. And I'm going to drive you to that one picture where we're actually measuring inside the endosperm. So we can see that using XRF, we actually have more iron in the endosperm of the mutants compared to the wild type. So we're very happy to see more iron in the endosperm of our mutants, but we also increased the resolution of what we could do. So one of Hudson's students was working in the Canadian synchrotron, and we sent some samples to him, and we did this kind of analysis of the embryo to match that was the one was done by other people previously. So we can see here the iron distribution in the embryo, in the wild type, and here in our mutant. So we can see that we have like this super bright colors. We have much more iron in our mutants. And this is consistent throughout all our lines. So this was cool, but we don't know exactly what's happening. We don't have enough resolution. So now we're using national technology. We're going to Sirius, which is our synchrotron. It's one of the best in the world. I really love that we have this equipment here in Campinas. And when we did that, we actually got this kind of data. So this is the low resolution. So it's 5 microns resolution in the rice embryo. You can see here what's going to be the shoot. This is going to be the root, and this is the scutellum, which absorbs what's in the endosperm. And here is the mutant. Oh, sorry. Here is the mutant. So we can see we have much more red color, which means more iron. Of course, we can separate those to make it easier for you. And you can see here much more iron in our mutant compared to the wild type. And we can focus in some of those regions, like the scutellum and also the plumule, which is what's going to be the shoot. And we get some higher resolution image, which is almost single. We can almost see inside the cell what's happening there. So here's the wild type in the scutellum. You can see much more red here, and here is the coleoptile. You also see much more red, which means much more iron in those regions. So iron is not only increasing as a bulk concentration in the seed, but it's also changing the distribution. So we're amazingly happy to see that this is kind of not working. Oh, there you go. Thank you. There you go. So, of course, we wanted to know exactly what's happening there. We did some sectioning. This is a partnership with another friend of ours that lives in Chile. He developed a technique where we can actually stain iron within cells. So we're doing that in the scutellum. This is not very good for you to see, but I have a picture here so we can see. This is the scutellum. You can barely see any iron inside it, but this is one cell of the scutellum. You can see this little bodies which accumulate. We think these are protein bodies accumulating a lot of iron in the scutellum cells in the mutants. That's very consistent across all of our lines. So this was done by Angie Sierra, which is a PhD student in our lab. She just finished her PhD. So we're very happy. Our double mutants actually increase iron in seeds, in the embryo, and also in the endosperm, particularly in the embryo, in what's going to be the shoot, and also in the scutellum. But you may know that rice plants also suffer from iron excess problems, so that can become very toxic when the plants are growing in these kind of fields. Now, the state where I live in Brazil produces 70% of all rice from Brazil, and Brazil is the biggest producer outside Asia. So, and everything there is planted this way, and this gets the soil very reductive. Iron becomes iron 2 and gets too much inside the plant. So iron excess is a problem. This is what happens with the plant. So we were worried that could be a trade-off. Okay, we biofortified the seeds, but we also get some sensitivity to iron toxicity. So the reason why we're thinking that is because those transporters are vacuolar iron transporters, so they do detoxify iron in physiological conditions. But VIT2, particularly the second copy, usually they are low expressed in the tissues where they are expressed, particularly in the leaves. But under iron excess, VIT2 is highly expressed. It's one of the top genes. This is very consistent across the literature in all the transcriptome data. So we were worried that these plants could be more sensitive to that one stress. And indeed, we decided to test that. So what happens when we do, this is our wild type plant, the 3 lines growing under control conditions, they were all happy. But when after 3 days of 5 millimolar, this matches almost what we get in the field when it's very heavy on iron. You can see that the mutants are more bronzed compared to the wild type. And when we do that for 7 days, they're much worse compared to the wild type. Of course, we have other physiological data to back that up. So this was really showing us a trade-off in biofortification. We increased something, but we got more sensitivity. Nobody's going to plant anything that has that problem, right? But I'm very interested in the mechanism. So we went back into the synchrotron, and we wanted to know exactly why the leaves why the plants are more sensitive, what's happening with those plants. And we decided to take a look inside the leaves. This is data from 10 days ago. We were here in Campinas. So this is preliminary. Please, this is just fresh out of, we got that figure ready yesterday. So it's the first time we're going to be talking about. So I'm very excited to show you if this works, of course. There you go. So what you're seeing here is in green potassium. Again, we're doing ionomics. So synchrotron X-ray fluorescence allows us to see 5, 7 elements depending on the tissue we're analyzing. So this is very helpful to us. So we can see here potassium, which matches exactly where our potassium is everywhere. So we can see our leaves. This is a cross-section of the leaf. And what you're seeing here, this is the xylem. There's three pieces of the xylem. Here's the phloem. And what you're seeing here is the border of the vasculature where the bundle sheet is. So you can see calcium in blue and iron is in red. So we see this ring of iron. You may think that that's actually the bundle sheet, but it's actually inside the bundle sheet. It's what we call the mastome sheet. This has vascular origin. So you can see that these cells are actually detoxifying iron because these plants are under iron excess. If you actually increase the view here, you can see this is probably one cell, and this is one vacuole that you're seeing here. So the reason, what we think we're seeing here is that the plant is detoxifying into the mastome sheet vacuoles using VIT2. The reason why we think that is because, of course, we have also the mutant. So we can see here the same image I just showed you. Here we can see another sample. We have more replicates of that. And here's the mutant. So when we have a mutation in both transporters, you cannot see the ring anymore. But if you play with the scale and you increase the points you're seeing in your background, what happens is that this one leaf has more iron spread around the mesophyll. So the model we were thinking is that there's too much iron coming from the xylem, and if you don't have VIT2 to detoxify that in the mastome sheet, you have more iron going into the mesophyll, and that is causing the toxicity that we see, the exacerbated toxicity we see in our mutants. So I think this is super cool that we can do that here in Campinas in collaboration with Brazilian friends. So we're very happy with those results. Of course, I don't know how I'm doing with time, but I'm almost at the end. So we're very stubborn. So we know we didn't get the best results with our plans, but we're trying to add more stuff to those. So we segregated out our transgene, and now we're adding at least another addition in another candidate gene, and we're also expressing some genes that can further increase the concentration of iron in rice. So we may get that at least in upland rice. It may work. So we're very stubborn in that sense. We're also we want to do that in other crops. I was talking to some people to do that in other crops as well. So we're very interested in barley, maybe some other. So I think that could work in plants that don't have problems with iron access. So I think we found something interesting that could work. So just to finish, if I have 1 minute, do we have 1 minute? Okay, thank you. I just wanted to show you, this is an extra. This is not part of the 2 stories, but I'm super excited about that too. We love, as Scott does, we love the wild relatives of rice. So for those of you who don't know, there's a lot of wild rice around. This is a pan-tropical genus, and now we have genomes for all those. So we wanted to know, if we want to do biofortification, we want to know maybe there is some species that already have solved the problem for us. Maybe they can accumulate a lot of iron in the seeds and maybe in the endosperm. So we decided to take a look at that. So we did one screen. This was actually during my Serra Pilheira years, so they sponsored that for us when I was just beginning. And we did, I think, one of the largest screens of seeds, the seed ionome of wild relatives. So what you're seeing here is that every column is one element. We measure all of those. And here you can see all the species that we did. That means almost 250 accessions measured and 18 of all the Oryza species, which is about 27. So we got all this data. Of course, I'm interested in my favorite element, which is iron. You can see here the variation depending on how close they are to Oryza sativa, to the rice that we eat. And we have, of course, at least one interesting candidate here, Oryza punctata. The species is consistently high in iron in its seeds. And now, really quickly, we're going back to the synchrotron to see where that iron is localized. I still don't have this data quite right, but you can see that indeed the embryo has more. We don't know about the endosperm yet, but we are pursuing that. We want to know exactly why the distribution is changing. So anyway, with that, I would like to change to finish. I cannot do anything without a really big network of collaborators in different parts of the world. Most people have always said yes to our ideas and to help us out. So there's a lot of people involved here in Nuspi, in my hometown, in Chile, also in Erie and Germany and several other places. So I'm very thankful to them. And of course, to the students of my lab and other labs, this was done by them. This work was done mainly by Angie. Angie was a PhD student in my lab. And with that, I would like to close with a take-home message. This is my daughter saying that we want you, students and collaborators and postdocs. If you're interested in what we're doing, we need more people in our lab, so we're hiring all the time. So please say hi, send me an email. I'm always open. Thank you.
Thank you very much, Dr. Hikashinevsky, for your insightful lecture and for sharing your valuable knowledge with us. And now, I would like to invite Professor Jamie Schnabel and Professor Douglas to join Dr. Felipe at the round table for our questions and answer section. A round of applause, please. We now have time for questions.
I really like the talk of all of you, and I have several questions, but I wanted to ask Felipe about what he thinks the advice for breeders would be, because we are talking about biofortification. I also work with sweet potato, and we are trying to make it double biofortified or triple biofortified with iron and zinc. And you show very early in the talk that it responds, well, it's something that the plant needs outsourced, right? It's not something that it produces like beta-carotene, right? So it's almost like you're breeding for that specific type of soil, right? Which is what genotype by environment is about, right? But it turns out, I was thinking or wondering what would be the way to go in terms of selecting individuals for making sure that they are going to be biofortified regardless of, well, soil. I don't know, how we can disentangle this in terms of breeding, like for breeding initiatives, right?
Oh, there you go. Oh, sorry. Yeah, well, you tell me, you know more breeding than I do, but to me, from the molecular biology side, I think what we're looking for, it's not that, okay? So we got, depending on the line, from 50 to 100% more iron. That's not gonna fly in the field. I have no hopes that's gonna work. So if we need something that, So if we want something that works not in all environments, but in several, it's going to be something that it's very strong. And I think we're going to have to do in a design approach. Like we know the mechanism, and we can change that, and then it might work in that way. That's what I think. But I have no hopes to get something that works everywhere. So one of the best ideas I've got recently is exactly that. We're using Nippon bar, which is a model cultivar, and it's for flooded soils. And so like, well, you try with upland rice, because upland rice is not as productive as lowland rice, but you may get high iron in that one. And if you have a strong enough effect that is like 3 or 4%, 3 or 4 times, maybe that could be somewhat consistent across environments. But might be just a vain hope that I have as a known breeder, and you may tell me like that's never going to fly in the field. I don't know. So sorry.
No, that's perfect. And a follow-up question is about how you think ionome could help in terms of understanding bioavailability, because this is another thing, right? The iron and zinc could be there, but they are not bioavailable. And for example, in the case of sweet potato, you know that it's more bioavailable than in case of potato. So it doesn't matter if potato has a lot, the people are not going to be able to get it from the potatoes, right? So this is something I was also interested in hearing from you.
Well, I think the approach that we did just to measure everything is not going to solve that particular problem, at least not with ICP-MS, which is a very bulky way of knowing the ionome, right? But we can get better using synchrotron or any X-ray fluorescence method, because we tried to do that and we didn't succeed. Sometimes, depending on the setup of the beam line, you can also see phosphorus in your sample. So at least, I don't know what's the case in sweet potato, I'd love to hear about it. But in rice, the problem is usually phytate, right? So we have phytate bound to zinc and iron. And if you get your iron more towards the endosperm in the seed, and you get your phosphorus still stuck to the aileron layer, to the external layers, then you're kind of fine, because that should be bioavailable. But you would only know if you tested. There's, of course, standardized tests like even cell culture and in murine models that you can do. We've never done that, so that would be a next step. But I would only spend time and money on that when I get something super high. Even if it's a model cultivar, as we're doing. Now we're going to try to do some local stuff, but I will not waste my time to know if 50 or 100% more is bioavailable. I think you need something super big. One of the differences, though, is that rice has no diversity for that. For zinc there's some biofortified rice, but not for iron. Iron is super low, the baseline is low, and everything is low. So that's why I think this is the case for genetic engineer, and that's, we're gonna have to introduce some variability there. So.
Good afternoon, thank you all for these amazing talks. They were really, really good. And I have questions for Dr. James and Dr. Douglas. Dr. James, with your work with Charlie, is it me? And the fungal microorganisms, did he get the chance to isolate any of those microorganisms?
So your question was whether or not we've actually isolated the microorganisms from the leaf surface?
Yes.
The short answer is no, we have not yet had a chance to do that. In 2021, those samples are long gone. No, this one.
Okay. Yeah. In 2021, unfortunately, those leaves were long gone before we realized this might be an option.
We have leaf tissue, so we're going to look directly for the DNA of the microorganisms. And I'm working with a very talented plant pathologist who can actually give us isolates of some of the same pathogens, so we can test with inoculation if we see the same response.
Thank you. And Dr. Douglas, I wanted to know why you chose to do GCMS for your metabolomics, since you'll be missing most of the secondary metabolites with that technique. And I think that would actually give you a more interesting and differential response in your analysis.
Yeah, you were right, but by that time, most of the facilities here in Brazil were delivering GCMS target mostly for primary metabolites. So that's basically what I had in my hand. So one of the major issues within my research line is exactly this. Okay, So I need to be a little bit deeper in terms of secondary metabolites, because there is where I'm going to find more diversity. That's not for this work, but for other works. I have collaborations abroad, basically working in delving into secondary metabolites.
Professor James, you know that artificial intelligence keeps surprising us, and you showed that having different tools capable to replace the humans. And do you still believe that the breeder's eye will remain indispensable?
That's a very good question. And those were the slides I kind of skipped over right at the end, was about seeing if we can learn a little bit about what the breeder's eye is and what people see. Because we know it's really powerful. There are studies that have looked at early stage field trials in Africa where you either have one small plot and you can either measure the yield directly, or you can ask farmers to come through and tell you which varieties do they like, which ones they don't. And then when you go on to large replicated yield trials the next year, you have more seed. The farmer's appreciation score is a better predictor of performance than the plot. And there's a lot, in one plot there's a lot of environmental variability. A deer or an antelope came through and took one bite out of the plot. It throws things off a lot. I think we are going to start to replicate the breeder's eye. I think we're starting to be able to figure out what's going on, which is that farmers and breeders have been trained over many years. What are the other phenotypes that are normally associated with good yield in their target environment? And some of those are much more stable. They have less G by E and less just sort of experimental noise than yield itself. That doesn't mean we won't need plant breeders. The two things that humans are going to be much better than AI for a long time are, one, knowing what are the right questions to ask. So maybe that's saying, oh, we need to go after iron biofortification in rice. The computer's not going to tell you that. It might give you some ideas about how to do it, but it won't tell you what to go after. The other, and even more important thing that the human does, the AI doesn't, is to critically examine the data the AI gives you, because it is wrong a lot. It's very much like a first-year graduate student. It will come in, knows how to do a lot of things. Sometimes it works. Sometimes you get a result that looks really good but is completely wrong. And one of the things all of us learn is that gut sense, not of the breeder's eye, but the scientist's eye. Something's wrong with this result. I need to dig into it. And I think those are the two things that are going to be really essential for all of us as scientists, as plant breeders, going forward.
In fact, this is not a triploid, it's a tetraploid arabica. Yeah. But I do agree, because with this vision, of course, that people look for rust resistance because it's a major trait. But I quite believe that other alleles related to a more fast response to stressors are being compared in these introgressive varieties.
And you are not able to disentangle the two subgenomes in terms of gene expression contribution, because you show like the results all together, right?
Yeah, I didn't take a look on this, but I do believe that we have some differences within gene expression. In this experiment, I did not put an eye on this, but usually when you look to, for instance, secondary metabolism, you do see a preference of one of those two subgenomes depending on the phase.
Okay, so thank you everyone for this amazing discussion in this section. And on behalf of the organizing committee, we would like to express our sincere gratitude for your presentation and for sharing your expertise with us today. And as a symbol of our gratitude, we would like to offer for each of you a small gift. So please, Professor Douglas, you can be the first one.